Okay. Um, Today we're going to talk about some molecular forces in biological micromolecules like uh, proteins or DNA, RNA, peptides. So when we study the biomolecules, in a lot of times we need to study some physics features because um, if you study the biomolecules in a very small scale, like in nanometers or angstroms, then most of the things they are physics. For example, there are some forces between molecules, energies between molecules. So today let's talk about the force and energies. Hopefully this can help you to understand the future classes like electrostatic force calculations, which will be demonstrated by Shengjie in the future. And also it's helpful for understanding the molecular dynamic simulation, MD simulation. Chitra and Yixin will demonstrate how to use NAMD to run the MD simulation. But let's try to understand some basic um, um, forces in molecules. So today let's focus on several different types of forces, the Coulombic forces, um, and some other forces like not force, but some effects like um, salt bridges and hydrogen bonds. So first of all, there are some different types of interactions between atoms in molecules, like in proteins. One of the most common interactions is a covalent interaction, or the chemical bonds. So such chemical bonds hold the atoms of biopolymers. For example, this is a classic folding process of a peptide um, folding into a protein, let's say. So a peptide is a sequence of amino acid. So let's say in this model, each beads or sphere represent amino acid. So those amino acids are connected by chemical bonds or covalent interactions or covalent bonds. Such bond is very stable. Let's say here, covalent bond energy are on order of 100 kcal per mole, and it doesn't break when water boils, when protein fold, when RN fold, when DNA fold. So it's very stable bond. For example, when this peptide fold into a protein, so a lot of interactions has been changed. For example, we can see, let's say this atom and this atom, they may have some interactions or forces, including, let's say, van der Waals force and electrostatic force. This force is changing during its folding. For example, when this protein or this peptide is folding in this stage or next stage or next stage, at the end, maybe this atom, these two atoms or this atom pair is getting, let's say, closer from this distance to such a very small distance. So the interactions of van der Waals or electrostatic interactions, they get significantly changed during the folding process. But one thing that doesn't change is the covalent interactions. For example, the neighboring beads or neighboring atoms, this one and the neighboring one, they are connected by covalent bonds. So this bond is too stable. It's difficult to break it or to change the distance between two covalent bound connected atoms. So these bonds they don't change during the whole folding process. The only thing change is the other forces, which is non-covalent interactions. So they are relatively weak. For example, van der Waals force and the electrostatic force. So they are relatively weak and usually with a free energy of less than 1 RT, or let's say usually it's less than 10 kcal per mole, compared to 100 kcal per mole. Um, this non-covalent interaction is relatively weak, so it's easily to be broken. But cumulatively, the energy can be huge. For example, if you have this protein folded, then it's very difficult to unfold it back into this form because there are too many non-covalent bonds like electrostatic forces, van der Waals forces. If you want to change this folded state into the unfolded state, you need to break a lot of non-covalent forces. So if you accumulate many of the non-covalent interactions like van der Waals and electrostatic force, the total energy could be very low. Still, the structure could be stable. Let's see. And such bond, sometimes it's within globular protein, for example, like this one. And sometimes not sometimes, it's always in globular protein, and it's also between protein and water atoms. And also it's in the protein-protein interactions. For example, if you have two proteins and they interact with each other, so inside the protein, it has a lot of covalent bond and non-covalent bond. But between the two proteins, A and B, you will have many non-covalent bond. Usually it's electrostatic.
Teacher says waiting.
Oh, okay. Yeah. Okay, thank you. So between two proteins, there might be a lot of non-covalent bonds, including some electrostatic forces and van der Waals forces. And for electrostatic forces, sometimes the residues which have electrostatic interactions, especially attractive interactions, sometimes they form some stronger interactions, which is called salt bridge. We'll introduce salt bridge later, but at least here you know in protein A and B, they may have a lot of non-covalent bonds. So each bound is weak, but if there are a lot, it could be strong. The protein is held between powerful forces and large force pushing the protein to fold. Okay, so this is a folding problem. So due to the lot of forces like electrostatic forces and van der Waals forces, the protein can fold automatically from a peptide into this format. So the origin of protein folding actually is due to the interactions or the mostly from the non-covalent forces. Examples of folding and unfolding. Okay, so because of the forces, the proteins in nature, the protein can fold from peptide into a globular protein. Some examples are shown here. Okay, so it's easy for protein to fold from peptide to a globular protein, but usually it doesn't unfold automatically because, as we said, you need to break a lot of forces or bonds so that it can unfold. Um, so when you heat an egg around 60 degrees, then the protein denature and aggregate um, into the egg white. So egg white, if you see the egg white, that means the protein has been denatured, and there's no way for you to change the egg white into the original protein format, which is transparent fluid format. Or another way is if you add some lemon juice into the milk, then your milk will be, the proteins in your milk will be denatured and aggregated. And you can see a lot of white stuff in the milk. And it seems like the milk is separated from water. So that's another example of, uh, um, let's say, denature a protein. Okay, now let's talk about some classic non-covalent interactions. So here in this slide, it doesn't introduce at all for van der Waals interactions. It's only focused on electrostatic interactions, but it doesn't hurt for us to maybe look at the van der Waals force just a little bit. So what is van der Waals force? For atoms, okay, let's go back to this, maybe this picture. So for some atoms, for example, let's say this atom and this atom, they're far away from each other. But no matter how far are they, there's always an interaction which is called van der Waals interaction. Usually we use V, D, W. Oh, it's terrible. So let me do it again. VDW. So van der Waals interactions, no matter how far away they are, there's always a van der Waals force between any two atoms. If they're very far, then this force is almost zero, but they are attractive. So these two atoms are attractive, but because they're very far, so the attractive force is very small. It's like zero. So when the protein is get folded, these two atoms might be getting closer. For example, here and here. So these two atoms now they're getting closer. So the van der Waals force will change. When the distance between two atoms are shorter, then the force becomes stronger, and it's still attractive force. But if you put two atoms too close, let's say very close or very crowded, then in this case, the force becomes repulsive force. So the two atoms will try to repel each other. This is the nature or feature of van der Waals force. If we look at um, the curve of the van der Waals force, the force against distance looks like this. So positive means repulsive, negative means attractive, zero means there's no force. So bigger value means the force is stronger. So the x-axis means the distance between two atoms. So you can see if the distance of two atoms is very far, so the force is almost zero. If they get closer, you will see the force is getting stronger in a negative way. That means they're getting stronger, but it's attractive force, because negative in this figure means attractive. And then at some distance, which is r0, that's the most comfortable distance for those two atoms. At this distance, the attractive force is the strongest. But if you push the two atoms to be even closer, then the negative force becomes smaller and smaller and smaller and then get zero. And if you try to put them getting even closer, then the force becomes positive, means repulsive. The two atoms will try to repel each other. The repulsive force increases quickly. So even you push them a little bit, you will figure out the repulsive force is getting very strong. So that's why in protein folding or in protein-protein interactions, you never see two atoms get clashed. For example, in protein folding, if you see two atoms are overlapping with each other or clashed, then that means the distance of these two atoms is in this range. They are repelling each other. So it's a repulsive force and it's very strong. So during the MD simulations or even other algorithms like docking or something, usually we avoid two atoms to get too close to each other due to the van der Waals interaction. So van der Waals interaction is very interesting. So it's common for every two atoms, no matter it's positive or negative atoms. It's not related to their charge. It's common for every atom. Even the atom is neutral, the two atoms they will have a van der Waals interaction. Um, and usually there are some equations to describe the van der Waals interaction. Do you know what is the equation for van der Waals forces? Okay. The equation for van der Waals, there are several different types of equations that describe van der Waals interaction, but the most popular one is Lennard-Jones equation or 612 equation. Let's see, it's too complicated. Lennard-Jones. Okay, so here is Lennard-Jones potential. So for epsilon is constant, sigma is constant, r is the distance between two atoms. So sigma over r to power 12 subtracted by sigma over r to 6. So that's the equation. In this equation, if you draw the if you plot this equation in xy coordinates, you will see this equation looks like this. It's very similar to van der Waals feature. So usually we use this equation to describe van der Waals equation. So even in MD simulations, we use this equation to calculate the van der Waals force between two atoms. So that's van der Waals force. Maybe we can go a little bit more or further about the van der Waals force. So now let's think about why two atoms may have some forces. You have two atoms atom A and atom B. So even if they're neutral, still they have van der Waals attractive forces. Why is that? Um, it's different from gravity. So for gravity, we know for two planets or two objects, um, they always have uh, gravitativity forces between them. But van der Waals is different. Van der Waals is another type of force. The origin for van der Waals force is induced charge. So let's say an atom, let's say if it's neutral, actually it's not uh, homogeneously neutral. Let's say each atom is composed by nucleus and electron. Nucleus, of course, is positive. Electron is negative, 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 negative. So in nuclear, it's positive, but the electron cloud is negative. So the charge distribution for atom A is like that. However, okay, so for atom B, it's the same. However, if you put A and B close to each other, what you will see is you will have some induced charge. I believe all of you have done some experiments in physics class. For example, you use a rubber band or rubber rod to scratch your hair, and then the rubber band can attract some small pieces of papers. So that's because of the induced charge. So the same thing here for A and B. So if A and B are getting closer, then the charge in A and in B, they will be redistributed. For example, if we consider um, the center of positive charge here and the center of negative charges to be here, this is the case for A. For B, it will be the same case because, let's see, when they get close to each other, the negative charge will attract positive charge. So the positive charge of B will prefer to be on the left, and the negative charge here will repel negative charge of B. So the negative charge of B will be on the right side. So that's induced charge. So that's very natural. So if you have two atoms or two objects, and they have some free charges which can move in the object, they will redistribute the charge in this way automatically. So that's the origin of van der Waals force. Now you see this is negative, this is positive, they will attract each other. So that's the origin of van der Waals. So here it has an even more intuitive picture to show the origin of van der Waals force. So the induced charge will attract each other. Okay, do you have any questions about the van der Waals forces? Okay, if you don't, now let's move on to Coulombic interactions. For Coulombic interactions or electrostatic interactions, that's more straightforward because we have all learned it in your college physics or in your middle school physics or something. If you have a negative charge and a positive charge, they will attract each other, and the equation is shown here. If they're in the same charge, for example, they're all positive, then they will repel each other. So this is the same thing in the protein. In protein, you may have some positive charges or positive atoms or negative atoms. So they will either attract or repel each other. Types of Coulombic interactions. Okay, the interactions of Coulombic interactions or electrostatic interactions are between charged atoms. also between charged amino acid. For example, if you are familiar with um, biochemistry, you will know there are 20. Okay, let's look at the table. That's even better. Amino acid. So if you are familiar with biochemistry, you will understand there are 20 basic amino acids. And of course, those 20 basic amino acids could have some modifications or changes in proteins, which result in much uh, more um, different types of amino acids. But here there are 20 basic amino acids. In different amino acids, they have different charges. For example, most of them are neutral, but some of them are polar. Polar means uh, totally it's neutral, but part of it is strongly negative. The other part is strongly positive. Um, and there are also 5 amino acid. Three of them are positive, which is arginine, histidine, lysine. For these 3 amino acids, they carry uh, one positive charge here, here. For histidine, it's more complicated. Um, Over 50% of the chance it's positive. It depends on the environment. In most of the case, it's positive, but not in all the case. For arginine and lysine, in most of the case, they're positive, unless it's in a very, let's see, unless if the pH is very high. But usually when pH is 7, they're always positive and they carry one electron charge, one positive charge. There are also 2 amino acids which are negative. The first one is aspartic acid. The second one is glutamic acid. So for these 2 residues or amino acid, they have negative charge. So if you go back to this slide, you may have some interaction. You may see some interactions between charged amino acid. It's mainly between either lysine, arginine, glutamic acid, and aspartic acid. So it's positive, positive, negative, negative. If you see these 4 types of amino acid in your simulation, if they're getting closer to each other, there will be some electrostatic forces between them. There are also interactions between protein charges and ions. So for example, if you have a protein, it may have some positive charge, negative charge distributed like that. Surrounding the protein, you will see a lot of water molecules like that. And also, there should be a lot of ions. So most probably, if here in protein it's positive, in your simulation, you may see some negative ions, which has been attracted by the positive charge of the protein. And here it might have some positive charge. So that's interaction between protein charges and ions, and also between protein charges and waters. So water is neutral. However, water is very special. It's quite different from any other gas molecules because water is very polar. So that's the Coulombic interaction. Now let's see salvation energy. There are 2 types of salvation energy. One is electric component of salvation energy. The other one is, or sometimes we call electric component salvation energy to be polar. The polar part of salvation energy, and B is the nonpolar part of salvation energy. Polar means you have positive and negative charges distributions. Nonpolar means you don't consider negative, positive charges. So polar means charged, nonpolar means no charge, no charges are considered. Um, so imagine that uh, you put some protein into water from the vacuum into water. You need some energy to transfer a protein or a molecule from vacuum to water. So part of the reason is when you move some proteins into water, let's say if you have, here it's simplified, it just give you an ion, but if it's a protein, let's say it's positive, negative, negative, positive, the negative positive charge, they will interact with water molecules, and that needs some energy. So this part of energy is polar component of sufficient energy. Okay. Another part is even if this protein is neutral and there's, let's say it's not a protein, even if you put a stone in the water or a piece of metal into the water, there's no charge distribution at all. It's neutral. Still, you need some energies to make a cavity because you need energy to break a lot of hydrogen bond between the water hydrogen bond network so that you can create a cavity, a cavity so that you can put your protein or other molecules into this space. So this is called the nonpolar uh, energy, nonpolar part of sufficient energy. Um, there are some amino acids which interact with each other. Sometimes if they are getting very close to each other, they form a salt bridge. For example, if a lysine is very close to a glutamic acid, as we described, lysine is positive, glutamic acid is negative. When they are very close, usually we set the threshold to be 4 angstroms. If the distance between those two charged atoms are less than 4 angstroms, we will say that's a salt bridge. Here, that's another salt bridge. Also, no, it's a hydrogen bond. So let's talk about the hydrogen bond later. So, uh, so for salt bridge, it's very common inside the protein, and it's also very common between two interacting proteins. And salt bridge helps the protein to be stable and help the two proteins to bind to each other stronger. Now let's discuss the hydrogen bond, which is another type of kind of force. So first, let's look at water. So water is very different from other gas molecules. So if we look at the gas molecules in the air, okay, so here is a table of the gas molecules. Nitrogen in the air, nitrogen is taking the like 80% of the whole air. Oxygen and other molecules, and also there are a little bit water gas molecule in the air, but it's the concentration is very low. But if you think about it, it shouldn't be in that case, because if you see uh, the molecules in the air like hydrogen, hydrogen is a very light molecule. Um, so the molecule weight for the hydrogen is only 2. So it's a very light. If you look at, let's say, nitrogen or oxygen, oxygen is kind of heavier much heavier than hydrogen. Hydrogen is 2, but for oxygen, the weight is, or the mass is 32, because a single oxygen weighs 16. So O2 means 2 oxygens, that's 32. If you write it down, you'll figure out O2, H2. The weight of O2 is 32. The weight of H2 is 2. Now let's see H2O. The weight of H2O is 18. So the mass is between hydrogen and oxygen. Actually, it's the weight is not special at all. If you put H2O into all these gas molecules, you will figure out it's not the most heavy atom. It's not most light atom. It's just a very normal molecule. But why water molecule is so different from any other gas molecule? All the other gas molecule, if you look at the tables, are all in gas uh, format in nature or in the room temperature. Only exception is water. So water in room temperature, it's a liquid. It's not a gas. This is because of its chemistry nature, because it's a polar molecule. If we look at the molecule of H2O or water molecule, it's oxygen and hydrogen. In oxygen, so the total or the whole molecule is a polar molecule. In the oxygen part, it's negative. In the hydrogen part, it's positive. And it's a very negative and very positive. Due to this nature, um, the, the molecule of water, they easily form hydrogen bond, which means the hydrogen can easily share its electron cloud with another oxygen. This oxygen is not its own oxygen in its own molecule. It's another oxygen in another molecule. That's called a hydrogen bond. When an oxygen and a hydrogen is getting closer, hydrogen is easily to share the electron cloud with the neighboring oxygen. So that's called hydrogen bond. Because the water molecule is so polar, so in nature, every water molecule will form the hydrogen bond with other water molecules. And the hydrogen bond uh, makes the water molecule attract each other much stronger than any other gas molecule. For example, if it's O2, it's oxygen molecule, it may get very far from each other. So it's in a gas state. But for water molecule, due to the hydrogen bonds, they have to be very close to each other, because that will reduce a lot of energy due to the hydrogen bond. So the water molecules, they have to be very close to each other, and then it's in the liquid state in nature temperature. So that's why in all the nature gas table, only H2O is in liquid. All the other gas molecules they're in gas state. So here you can see how common is hydrogen bond. It's in the water, it's in the protein, it's in many molecules. If there's a hydrogen and if there's a oxygen, if they're getting closer to each other, very easily they can form hydrogen bond. And we call this hydrogen to be donor, and we call this oxygen to be acceptor, because the donor kind of donate its electron and share it with the oxygen. So the hydrogen bond between two water molecules, usually it's 1.8 angstroms. This is a stick presentation of water molecules. This is a sphere representation of two water molecules. So it's very common. For example, this is DNA. We know DNA, they have base pairs. Why they have base pairs? For example, A has to be paired with T, G has to be paired with C. The priority reason is the hydrogen bond, because when A and T, they match each other, they can form two hydrogen bonds. And when G and C, when they pair each other, there are three hydrogen bonds. By the way, it's not necessarily hydrogen bond to be formed by oxygen and hydrogen. It can also be formed by hydrogen and nitrogen. So you see this example, hydrogen and nitrogen, they can also form hydrogen bond. So due to the hydrogen bond interaction, the base pairs has to be paired in this way, ATCG in DNA. This is called the DNA specificity code. It's also very common in protein interactions. For example, there are three proteins bound together. If you look at the details, you will see there are a lot of residues at the interface forming hydrogen bond, which makes them stable. Okay, so here are some more details about hydrogen bond. And the hydrogen bond can also stabilize a protein. So for example, you have a protein which is formed by peptide, which is very complicated. It's like a mass. However, it's not, because nature choose this shape because in this shape, it gets a lot of bond, including hydrogen bond and sound bridges inside the protein. For example, there might be a residue here, another residue here, they form a hydrogen bond. A residue here, a residue there, they form a sound bridge. So finally, that's the nature state of folded protein. So this is water. We have discussed it. Hydrophobic effect. Okay. Hydrophobic effect. So let's see, in water molecule, every water, let's say this is a water molecule. Let's say this is oxygen, this is hydrogen, hydrogen, here and here. So for each water molecule, you have one oxygen and two hydrogen. All of these oxygens and hydrogens, they're forming sound bridges. If you put some object into or immersed into the water, it has to break some of the hydrogen bond. So that's the hydrophobic effect. If you put two objects into the water molecule, into the water solution, the two objects, if they are natural, then they prefer to bind with each other, because when they bind to each other, the surface of the two objects is getting smaller than the situation when they are separated. So they can break fewer hydrogen bonds. So this is called hydrophobic effect. Implication to protein folding. So all of the forces, they are considered, or they are in the protein folding. When the peptide folds into a protein, for example, in unfolded uh, state, the peptide may have different format or different conformations. It's changing from this format or conformation to the other conformation to this conformation to this conformation. It's kind of uh, moving or uh, changing the states from one state to another state. And then while it's folding, the energy is getting lower and lower and lower. And at the lowest states, you get the native structure of the protein. In the native structure of the protein, it doesn't have so many different conformations. Right? Usually it has only one conformation, which is the lowest energy state of the protein or the native structure of the protein. And during this folding process, all of the forces are functioning. For example, the van der Waals force, the electrostatic force, hydrogen bound, salt bridges, they are forming and break, and then form again and break again. And finally, the nature will find the lowest energy state. In this state, you have the lowest energy by generated by all the forces. So that's the forces in protein folding. Okay, so that's all for today's class. We introduced a little bit about some forces in protein folding, but it's not only for protein folding. For protein-protein docking or protein-protein interactions, you have similar forces. Okay, that's all for today. Do you have any questions? Okay, so if not, I will stop the recording.